Hello, everybody, and this is our microarrays lecture, the second one and the last one that will go towards the second midterm next week. It's going to be a fairly quick one. We will cover the basics of the microarray. Microarrays, we'll talk about how the microarray chip is being made, how the nucleotide sequences are being added to it, how the experiment is being conducted, and eventually how the results are getting analyzed. A lot of this will be familiar to you, and the reason being is that microarrays has essentially the same goal as the RT-PCR, and that is studying the gene expression profile, how different genes respond to different conditions, or maybe how different cells of different tissues, which genes they activate and which genes they shut down. The main advantage of this technique, the microarrays over RT-PCR, is that microarrays allows you to study multiple thousands and thousands of genes at the same time. So you don't have to set up individual PCR reactions for each individual gene that you're looking at. So the core concept behind the microarrays is going to be exactly the same, and these slides are familiar to you from the previous lecture. Not going to spend too much time on them. You have different types of cell, different tissues. You have the same genes, the same genotype in all of these cells, but depending on which genes are on and which genes are off, you have development of different phenotypes, different cells with different physiological properties. The same applies to cells that generate diseased phenotype and the one that express healthy phenotype. The same genes, but different expression profiles. So by looking into different differential expression of these genes under different conditions, you can potentially pinpoint what the problem is. So which genes are responsible for that phenotype, and if you can possibly in the future shut that gene down, you can fix the condition, some sort of a genetically predetermined condition. Or you can look into the mysteries of the developmental biology and see how different cells become what they are based on the expression profile of different genes in its genome. So same principle on certain genes, certain genes off. You're looking at them and you are trying to pinpoint which genes are responsible for the generation of the phenotype. One thing, don't forget for this discussion as well, we mentioned this in the last lecture. Keep in mind that any technique that relies on levels of mRNA on making the decision about the phenotype of the cell or the tissue, these techniques have to be taken with a grain of salt. You can actually add here one more arrow, and the protein should lead to a phenotype, right? So all of these techniques that rely on mRNA, like RT-PCR or microarrays, should be taken with a grain of salt because you are using intermediate molecule, mRNA, to make a decision about a phenotype. So you're kind of skipping a super important molecule, which is the protein, which is an effector molecule, so the molecule that is actually doing all the work, right? So you can have high levels of mRNA, as I mentioned last time, and you can have low levels of the protein. In some cases, levels of mRNA may translate into the same levels of the protein in a 1-to-1 ratio, but it's not always the case. The cell controls for the mRNA levels, and just finding out that the mRNA has high concentration at a specific stage in the life cycle may not be sufficient to make accurate decision about the generation of the phenotype. If that mRNA is getting destroyed, or if the protein is unstable or getting destroyed as well upon production, that means the phenotype is not going to develop regardless of how much mRNA is being made. But nevertheless, it is a pretty decent technique, very good actually technique for screening purposes when you are trying to narrow down the pool of thousands and thousands of genes that you have in your body, when you're trying to narrow that pool down to only specific ones that are probably worthy of a detailed analysis and protein work eventually. So instead of looking at all the thousands of genes, you just pick, based on these techniques, based on the mRNA levels, you pick like 5, maybe 10 of them. You significantly reduce that pool. And then always keep in mind that any sort of mRNA work has to be followed up by the protein work. If you can't show the link between the connection between the protein and the phenotype, any of the mRNA work is kind of useless. So, like I mentioned before, the major benefit of microarrays over RT-PCR is the fact that you are dealing with multiple genes, hundreds and thousands of genes at the same time in one run. So that significantly reduces the resources needed, the time that is needed to set up the reactions, and obviously allows you for a completely different level of analysis of your tissue, of your cell, of your condition. And one definition I want you to know for the test is the transcriptome. So the transcriptome, as the name suggests, you should be familiar, should be able to dissect it. So transcript deals with transcription, and ohm usually refers to large collections of the biomolecules, right? Like proteins, proteome, or transcriptome in case with RNA. So transcriptome for microarrays is defined as the complete set of transcripts, right? So all of the mRNAs that the organism has generated under specific conditions. Conditions could be exposure to certain stress factors, could be exposure to certain shock factors, could be just diseased versus healthy phenotype. Those would be different conditions. Or you could be looking at, if you're working in developmental biology, you're looking at just two different cell types, nerve and muscle tissue. So those are different conditions, and you are trying to establish how does the transcriptome, so the collection of all the mRNA molecules in the cell, changes how is it different in different cell types. Okay, so you are looking at all of the transcripts at once, which is, like I said, a huge benefit, saves a lot of time. So thousands and thousands of genes at the same time. And once you've seen this slide before as well, so once you have your data, once you've conducted either RT-PCR or microarray analysis, you can identify which genes are on and which genes are off under different conditions, say obese, non-obese tissue. So having this profile allows you to narrow down your search for the most interesting genes to only select a few. In this figure, and this is how you should be analyzing your genes, and that's how the microarrays work, you basically look for the genes that show differential expression. So in other words, you're looking for the genes that are either on in diseased phenotype and off in the healthy tissue, or the other way around, off here, on there. So any sort of differential expression. And then you can make a hypothesis that maybe potentially these kind of genes, like these two here, could be involved in the development of the phenotype. It's not a definitive statement yet. You still have to show that it's the case. You have to show that that mRNA that is being made, for example, from the gene that is on here and off there, that this mRNA is not getting destroyed completely, and at the protein level, at the protein level, a second gene has exactly the same condition, has exactly the same state as the second gene here, despite of mRNA being made. And because of that mRNA getting destroyed, you're still getting no protein here as well as there. So there could be no real difference between the contribution of the second gene to this phenotype, even though you have this gene on here and it being off there. So again, protein work is super important. But that's the second step of the analysis. And the first step, you at least want to identify, out of all the thousands of genes in our body, you want to identify which ones are worthy of any further analysis. Keep in mind also, I might have mentioned this last lecture, that the fact that both genes are on and are making mRNA doesn't mean that these genes 100% have nothing to do with the development of the phenotype that you're looking at. They wouldn't be analyzed in the first place, genes like this first one, which is on in this case as well as a non-obese tissue. It could be that despite the gene being on in both cases, the actual amount of mRNA that is being made in both cases could be significantly different. And that may also, it's a possibility that it may be reflected in a completely different levels of the protein that is being made in the first case and in the second case, in a diseased tissue and a healthy tissue. So in some cases, genes like that do not show the differential expression profile are actually important in the development of the condition. So the fact that both are on doesn't mean that this gene is 100% not involved in the generation of the phenotype. But you definitely would want to look at this gene in the second place. Like you wouldn't make a priority of this gene. But keep in mind the actual levels of mRNA, and which is even more important, the actual levels of the protein to follow are also important. So now that we're done with this background, let's look at the technique itself. So how the chip is being made, I'll have a separate video in a second. But before that, I want you to show what it looks like and what's the principle behind all of these spots on this chip. So the chip is usually made of glass, several centimeters squared in size. They're getting smaller and smaller these days with the development of the technique and different tools that we have. But each each microarray slide is separated into these multiple thousands and thousands of unique individual spots. So here under magnification glass, you have these 4 spots. The whole glass contains about several thousands of spots. So for each of these spots, on each of these spots, you have single-stranded DNA molecules that are being attached to this spot. And you have multiple copies of these strands. So 2 most important points to take home. Each spot represents a single gene. And if you look at this figure, you can kind of tell that each sequence is the same. Within the same spot, you have each nucleotide color-coded. You have brown, yellow, purple, green, 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 and then exactly the same sequence in this strand, brown, yellow, purple, and 3 greens, and exactly the same here, exactly the same there. So within one spot, you have exactly the same gene, a stretch of that gene, and this DNA sequence is a single-stranded sequence. That's a second major take-home point you have to consider. Next spot has, as you can tell by looking at the colors of these nucleotides, next spot has a completely different gene, the sequence of that gene that is being analyzed. It's a different pattern. You have 2 greens, then purple, 2 browns, and then yellow. And then again, all sequences within the same one spot have the same sequence, have the same nucleotide sequence. So all these 4 are having the same sequence. The number of nucleotide, a number of single-stranded DNA strands within one spot, obviously is more than 4. 4 is just an example, just a representation. It's a lot more than that. And then the third spot here has a unique sequence, as you can tell. The sequence is different from this one as well as from that one. And within the same spot, you have the same sequences, multiple copies of the same sequence. And then the last spot follows the same trend. So make sure you understand this is basically the foundation for understanding the microarrays. Single gene and single-stranded copies of DNA that are being attached to each of the spots. And now that we know what it looks like, let's watch a quick video about 5 minutes how the DNA is being synthesized on this spot.
The way you can do it is using one of these interesting devices. These are DNA microarrays. They come in different flavors and forms. They're made by somewhat different technologies and by different companies and academic laboratories. I've picked one here that comes in a very nice package. This has a little sliver of glass, a little square of glass, in which, in those little squares that you see on the slide there, each square has a different DNA sequence. There's a specific 25-letter DNA sequence in that square, and a different 25-letter DNA sequence in that square, and a different one in that square, and a different one in that square. Every one of these has whatever DNA sequence you would like to specify. You could type them in, and someone could make a DNA array that had different 25-letter sequences in it. How in the world could you do that? Now, I suppose you could go to the chemical laboratory and synthesize the first one, and then come and stick it down, then synthesize the next one and stick it down, and the next one and stick it down. But actually, the way they do this is to actually do it in parallel. They do it the same way that people make microprocessor chips in Silicon Valley. They have a mask, they shine a light on the glass. Where the light shines, the surface is de-protected, and you can wash on one of the DNA letters. You then reprotect the surface. Shine a light through another mask, deprotect certain spots, and wash on the next letter. Shine a light through a mask, wash on another letter. After 100 such masks, you could build up an average of about 25 specific letters at each spot, and depending whether on each mask you had black or clear, you could either activate or not activate each spot and build up a specific sequence of DNA letters in each spot.
So now that you have your chip in place, you have thousands of genes being spotted as single-stranded molecules on each of these spots. Obviously, the location and the sequence of each spot, as well as the location of each gene, is recorded in a database, so you know exactly what each spot corresponds to, what kind of gene corresponds to. So now that you have that information, moving on to the actual experimental design. So what is it you need to do in order to study the expression profile of your cells? And going back to this example with a beast non-beast tissue, it should be one thing it should be intuitive to you, I guess, by just looking at this figure. The fact that you have single-stranded DNA molecules attached to each of these spots should tell you something right away. And that something is that the whole assay, the whole microarray technique relies on the hybridization between the single-stranded DNA strands on the spot and the single-stranded DNA molecules coming from elsewhere. And that second set of molecules, single-stranded molecules, will come from your tissues, your control, as well as your understudy tissue. So what they do is they isolate the mRNA, and jumping ahead a little bit, since we started talking about the isolation of mRNA, this next slide describes how this is done. You just need to know the basic principle that mRNA has distinctive poly A tails, and we can use these poly A tails to isolate this mRNA from the solution by using the beads that have poly T residues, which will complement the poly A tails and therefore will help you extract the mRNA with poly A tails from the solution. So that way you end up washing away all the ribosomal RNA as well as the transfer RNA, and then you end up having just your mRNA. So make sure you know that. Make sure you know that for the test, the fact that you have poly A tails and then poly T tails on the beads of the matrix, stationary matrix. Once you have your mRNA from the tissue extracted, right, you have to convert it to cDNA. You have to convert it to cDNA, and one more step, characteristic of this technique, you have to label them. You have to label that cDNA with usually two types of dyes. The one that emits back in the green spectrum, it's about 570 nanometers, and the one that emits back in the red spectrum at about 670, I believe, nanometers. And the dyes that have been employed here, they're called Psi3 for the green one, and for the red one, they're called Psi5. So once your cDNA is being created, each of these cDNA molecules is being, first of all, remember what the cDNA is, right? Complementary DNA synthesized based on the mRNA sequence using the reverse transcriptase enzyme. Remember those. And once you have these cDNA molecules created, synthesized, you make them single-stranded, and you also label them, fluorescently label them with these Psi5 and Psi3 probes. So now the idea is that all of the cDNA that came from the diseased tissue sample will be labeled red with the red dye. And all of the cDNA that came from the healthy tissue or a control tissue will be labeled with the green dye. And what will happen next? So the next step of this procedure, this is the generation of the cDNA using the reverse transcriptase. We discussed that. So you have a probe that it labels the one set of mRNA green, the other one labels it red. So the next step, once you have your mRNA from the control and experimental setup synthesized. Make sure they are single-stranded. These single-stranded molecules labeled with a probe are being added to your chip, microarray chip. You flood the chip similar to how you flood the hemosiderometer. Just add the solution, it floods the whole surface. And what will happen is at each of these spots, you can kind of see these spots here in this figure, each of these spots carrying unique sequences, unique identical sequences within the same spot. Sequences of cDNA coming from the gene of interest, and then your cDNA, single-stranded cDNA, labeled with either red probe or the green probe, will start looking for the sequences that they are complementary to. So this, let's say gene number 1 will have the cDNA coming from the tissue sample, whether diseased or control. In our case, as you can tell by the color, the fact that it's red means that it came from the diseased tissue. So this gene number 1, the cDNA from that gene will find the complementary sequence on this spot number 1. And once it finds it, because of the complementarity, the hydrogen bonds will get established, and the single-stranded DNA from the tissue will anneal to the single-stranded cDNA that was synthesized on this spot, and you will have a double-stranded DNA formed. For the second spot here on this figure, you have exactly the same idea. This kind of dark backbone is your cDNA synthesized on the chip, and then this green stuff is cDNA coming from the sample. So for this spot, you will have all green labeled cDNA molecules, oligonucleotides, anneal to the initial core cDNA sequence. Once you shine the laser, once you shine the light on these spots, you will be able to see the color. For spot number 1, you'll have all red, a strong red signal. And for spot number 2, you'll have strong green signal. And then for this spot here, number 3, what might happen here is that you will have 2 types of cDNA, one labeled green and the other one labeled red, anneal to the sequence, the core sequence sitting on the spot. And first of all, why is this happening? Why are we having only red labeled molecules, cDNA molecules, anneal here? Why do we have only green labeled molecules, cDNA molecules, anneal here? And why do we have both types anneal here? So the answer is pretty simple. Each core sequence corresponds to a specific gene, right? So depending on which gene was active at different conditions, if you go back, for example, for gene number 1, let's say this one was gene number 1, it was only active in obese diseased phenotype. So only for obese tissue the cDNA was generated. Nothing was created here. So if that's our gene number 1, then if you look at this spot here, only red labeled molecules will be present. cDNA molecules will be present in the solution, and therefore only red labeled molecules will anneal to the sequence, the gene sequence that is being synthesized, that was synthesized here. Why? Again, because only cDNA of this first gene, we labeled as gene number 1, only the cDNA in the obese tissue has been created, because only in this tissue the gene was read, was transcribed, and mRNA was made. Nothing was made here. And since all non-obese or control tissue cDNA is always getting labeled in green, then the only sample present for the interaction in spot number 1 will be the one labeled in red. For this one, exactly analogous to what we just talked about, the only difference is that this second gene is probably, going back to this figure here, the second gene would probably be this one here, the gene that is only getting transcribed in control conditions, and only the green labeled molecules are being generated therefore. Nothing is being created for this gene, nothing is being transcribed here for the obese tissue, and therefore the only type of molecules present for annealing are the green labeled ones. For this case here, as you can see, both types of molecules present, and that would correspond to this gene here, on in both under both conditions, obese and non-obese. So you're generating both types. Each type is labeled differently, red and green, and they will be given that the relative quantity of these cDNA molecules is approximately the same between two tissues, roughly 1-to-1 ratio, then you will have roughly as many green labeled cDNA molecules as you have the red labeled cDNA molecules. Once you flood the chip with solution with your cDNA coming from the tissue samples, you'll have both types and you'll hear, and therefore the signal that will be generated by the spot will be yellow, as the color code suggests. It's not going to be green, it's not going to be red, because once the green and red kind of superimpose on each other, you get the yellow color. You register the yellow color. Lastly, this spot here, you have, as you can see, there is no cDNA bound, and you only have these stretches of the core cDNAs that were synthesized for this spot present. These stretches would be these ones here, the ones that you chemically synthesized for each spot. For this gene here, whatever that gene number 4 is, there were no green molecules, no red molecules present in the solution. Why is that the case? What is the molecular event that would lead to this kind of scenario? That molecular event would be when the gene is shut down in both conditions. Obese, non-obese, disease control. In both conditions, the gene is shut down. We don't actually have this kind of a condition here in this figure, but you can kind of add one more here, number 4. It would be off here, and it would be also off there. So no red or green is being produced, and therefore this spot that corresponds to that gene has no substance, no cDNA to anneal to. Therefore, the color that is being generated for spots like that would be black. There's no fluorescence whatsoever, neither green, nor red, nor yellow. So at the end of the day, so that's pretty much the whole technique. At the end of the day, what you will have is you will have to scan your chip. You would put it into a fancy machine, just similar to how the 96 well plate readers work. Shines the light, laser shines the light on the chip, and single spots that have different cDNA molecules annealed to them will emit the fluorescence back. And what you will see is, I'll jump a few slides ahead, what the real picture will look like is something like that. You have thousands of spots, hundreds and thousands of spots. Each spot here represents a single gene. And as you can see, you have a whole variety of different colors here. You have well-pronounced red, like this one here. You have well-pronounced green, and you have well-pronounced yellow ones. Let's say this one here, well-pronounced yellow ones. So for genes, and remember, you know the sequence of all of these genes, you know exactly the identity of these genes, and you know the order of these genes on the chip. So by looking at this gene here, this is a good yellow as well. Looking at this gene here, you can make a conclusion that for this gene, this gene was expressed both in healthy and diseased phenotype in both conditions, because the color generated is yellow. Therefore, you had approximately the same amount of red labeled and green labeled with Cy5 and Cy3 probes. The same amount of cDNA labeled with those probes present in your tissue sample. For the well-pronounced green ones, you know that it's for this specific gene is being only transcribed at control conditions, non-disease conditions. This one here as well. For the red ones like that, you know that this gene is getting preferentially expressed, transcribed in diseased phenotype. The one that in our example was a obese tissue because of the red cDNA being made. So now you can look at all of these genes. Now you can look at all of these genes and you can make a profile. You can, what people do, the next step, people make sense of this picture. But before actually we move on to that, there's one more type of spots that you have to understand. It's actually a whole variety of different colors. As you can see, it's not just pure green and pure yellow. For example, spots like that, they're not necessarily or maybe this one is a better one. This one here is not necessarily green, is not necessarily red or yellow, right? It's kind of orange. Um, this spot here is not necessarily pure green, right? This one here probably has a slight touch of yellow. This one is not pure yellow either. It has a little bit of red in it, and more red than green. So for orange spots like that, you should be able to guess what's happening here is that you have both. The gene is getting expressed both in diseased and non-diseased conditions because you are generating kind of yellow phenotype. But the fact that it's actually orange means that you have more red than green. So that means both genes are getting expressed in both. The same gene is getting expressed in both conditions, diseased and non-diseased control. But you get slightly more of the red labeled cDNA being produced for this gene. That means that refers to the scenario when I told you that even if both genes are on, it doesn't mean that you always generate the same amount of cDNA for both conditions. So for those orange spots, you have slightly more of the cDNA being made from this gene in your disease phenotype than you have it being made in your control phenotype. And obviously, you don't just qualitatively analyze your results, you analyze them quantitatively. So people link the intensity of the color or the maximum absorption of that light and with the amount of the cDNA that was made. So the more orange is the spot, the closer it is to red, the more red DNA you have. So the more gene was getting expressed in diseased phenotype. And you can quantify it, and then you can actually put the numbers behind the levels of the mRNA based on the intensity and the maximum absorption profile of each spot. And that way you can say how much gene was getting expressed in disease condition and how much gene was getting expressed in your control conditions. So once you have a picture like that generated, so the previous slide, the one that I skipped, basically discusses that. Don't forget what the black spots here stand for. They stand for when the gene is active in neither of these conditions. So again, don't just blindly memorize these answers. Make sure you understand the logic behind it. You have yellow for when you have the same amount of the mRNA and the cDNA being made, and the black ones where you have no fluorescence. That means neither of these conditions made any mRNA, and therefore no cDNA was created as well. And yes, the yellow ones as well as the black ones would be not interesting conditions because you don't have any difference between the levels of the cDNA for these spots. Here, both are absent. Here, both are present at the approximately the same levels. The closer this color is to red and the closer it is to green, the more interesting it gets. Because the closer it is to green, that means the more there's a higher level of transcription of this gene in under control conditions, non-diseased phenotype. And the closer this yellow is to actually orange and red, that means the more of the red cDNA you have in place, and that means the higher is the level of expression of this gene under diseased condition. So that's pretty much the whole technique. That's how you analyze your microarray. The same idea here. There's no new information here. The next step, what people do, watch this animation. I don't think it adds anything new to the discussion that we just had, but one more thing that you need to know. I'm not going to be asking about this on the test, but once you generate the set of results like that, people try to make sense of what they see. So what they do is they try to group up all the genes that are getting predominantly expressed under control conditions as one group, and then all the genes that are getting predominantly expressed in diseased phenotype as another group. And then they try to make sense of these groups, and they try to come up with subgroups, and maybe the ultimate goal is to not just make the chip kind of look pretty, all greens on the left and all reds on the right kind of thing. No. What they try to do is they try to potentially establish the groupings of the genes. So the genes that are potentially working together in that phenotype, whether it's a healthy phenotype or whether it's a diseased phenotype. And that way people can, based on this data, they can come up with the new groups of the genes and therefore the new genetic networks that might be responsible for the generation of a specific phenotype. So you can find a whole bunch of genes that work together and potentially are responsible for the generation of a specific phenotype. Once that data has been generated, like I was saying before, the next step would be the analysis of your data on the protein level. I think it's on the next slide here. So once you have your microarray data, is that sufficient? The main question is whether it's sufficient to claim there's a link between the gene and the phenotype. The answer is no. We discussed this explicitly. Not going to spend any more time on it, but definitely no. It's a guaranteed question in the test. It's a very common misuse of this technique out there when people publish results based on the microarray data or RT-PCR data, and then what happens is, a couple of years later, or even faster than that, people post another publication that claims that the previous results were completely nonsense. The relationship is actually the opposite. So that's what happens when people are relying exclusively on mRNA data. It's not sufficient because the levels of mRNA is a very unstable molecule, and the fact that you have a lot of mRNA doesn't mean that you have a lot of protein. So always keep that in mind. Technique is getting cheaper and cheaper. RT-PCR is very cheap these days already, so people are very likely to abuse it and to use it to draw conclusions that are not really there, or the links that are not necessarily there yet. This next figure in the next slide kind of summarizes one of the major steps that are regulating, what are the major conditions that regulate protein function. So you can have mutations that may result in non-functional protein product. You can have a huge factor here. You can have alternative mRNA splicing processing, so when the same gene creates multiple different mRNA molecules, depending on the conditions. And then all sorts of downstream modifications of mRNA itself. So they can be sequestered away from where they need to work, they can be moved around, and they can be degraded before the protein is actually being made, especially with mRNA being unstable that it is. It is a very common mode of regulation. And then ultimately, once the protein is made, even if your mRNA got converted to the protein, let's say 1-to-1 ratio, whatever, or you know exactly what that ratio is, and you can convert, therefore, the levels of mRNA into the levels of the proteins eventually, so that issue is out of the question, but you still don't have the full control over that link between the gene, ultimately, and the phenotype that is caused by this gene because of chemical modification that may occur on the protein. So you can have some molecules affected by the modification that will increase the activity of the proteins, and some proteins will not be affected by that molecule. So that level of modification may have a huge impact on the activity of the protein. So just knowing the quantity is not enough. You need to know what kind of state that protein is in. And obviously mRNA analysis, even mRNA analysis by itself, as well as knowing the total levels of the protein, doesn't give you that information. Not to mention that a lot of complex phenotypes, which is again completely not accounted for by any sort of mRNA technique. Different phenotypes, especially the more complex ones like cancer and similar ones, they are not just being affected by one protein. They can be affected and almost always are affected by multiple protein products from different genes. So, and then again, each of those proteins is subject to all of these kind of modifications and regulations indicated here. Different proteins from different products may have different level of activity depending on activation or deactivation by the small chemical molecules. You can have different, not just modifications of that protein, but also small chemical molecules that will just attach to the protein, will serve as a cofactor, and will modify the activity of that protein even additionally. So none of that information, right, it can be collected from the mRNA analysis, and every single piece of that data is exceptionally important in establishing that accurate link connection between the gene and the phenotype, the actual condition that you're trying to analyze. As for major applications, I already mentioned that before. Make sure you know this list is, I think, yes, 5 major points. And we talked about the tissue-specific gene expression. So depending on different types of tissue, you have different genes being expressed, and therefore different properties are being imparted in the tissue. Developmental genetics is huge when you determine which genes are getting switched on in the early childhood and development of the fetus and so on, and that can allow you to replicate that pattern of development if you know exactly which genes are involved. You can track also how gene expression changes over time. For example, with this famous example when the lactose gene, the one that is responsible for making the enzyme that allows you to digest lactose, it actually gets switched off in some people later in their lifetime, and therefore people are incapable of digesting lactose. Genetic disease is huge, especially for the future applications once we are allowed, or once we are competent enough, I guess, to modify human genomes. You will have genetic tools which allow you to delete the genes, or maybe switch on the genes where they're switched off for all the wrong reasons, silence the genes, and so on. But before you do that, you need to obviously identify which genes are responsible. So any sort of mRNA analysis allows you to at least select, right, the major application for all of these applications. The major role that the mRNA analysis techniques play is the screening, the initial screening that allows you to at least identify which genes are involved, and maybe make some discoveries, as I was mentioning here, as to which new genes could be responsible in generation of that phenotype. Let's say if you group up all of these genes here into say 2 categories: the ones that are getting the ones that are getting preferentially expressed in control conditions, and the red ones, the ones that are getting preferentially expressed in disease conditions. Let's say you have, I don't know, 500 genes on the green ones, and then 500 red genes. But it might be the case that before you ran the study, you actually identified like 480 genes that you knew were switched on in control conditions. So by making these groups, by analyzing the microarray data, you might come up with new genes that might be a part of this genetic network responsible for a control condition or responsible for a disease condition. So identifying new genes that you haven't looked at before. Another major application, we will have a whole big lecture dedicated to it, studying the complex diseases and specifically identifying the genetic markers. The genetic markers, or I think I had it somewhere here, the so-called SNP profiling. We'll have a separate lecture on this for the final exam. Basically, this approach allows you to identify not necessarily the mutations that are directly responsible for the generation of the diseased phenotype. They're directly involved, right, in that phenotype, but allows you to find the markers, so the genetic elements, sometimes just single nucleotides in your genome, which serve as if you will, as road signs. Meaning that if you have that marker in your genome, you are potentially predisposed to a certain condition. That marker, if you don't have that marker, on the other hand, your chances are significantly lower to develop that condition. So the marker itself is not involved directly. It's not directly responsible for making, let's say, a faulty misfolded protein, but it just serves as I said, like a road sign. If it's there, then you should be aware that you have, let's say, 50% higher chance of developing this condition. Are you absolutely guaranteed to develop that condition? No. Because development of any phenotype depends not just on genetics, but also on environment and all sorts of complex factors that are coming together with environment, your lifestyle, your diet, and so on. But at least you can somewhat quantify right, your risk of developing a condition by identifying those genetic markers in your genomic DNA. And microarray analysis under high stringency conditions allow you to identify those sequences. We'll discuss that in one of the later lectures. Lastly, studying the pharmaceutical agents, all sorts of pharmacological agents these days. As you know, different people respond differently to different chemicals because of the different uh, genome expression profile of that genome. Some people make more of certain enzymes, some people make less of that enzyme, some people are completely devoid of that enzyme, as it was the case with the lactose intolerance, as I mentioned before. So some people are responsive to pharmaceutical agents, some people are not. So in order to study that, you study the gene expression patterns in response to a specific pharmacological agent, and then you look for those genes that are responsible for, let's say, metabolism of that drug or excretion of that drug, or maybe deactivation of that drug, and so on and so forth. And that can lead to corrections to the dosages that are being that are being supplied to a patient. Maybe will require modification of the chemical to make it less toxic to that person that is lacking a specific enzyme. So all sorts of studies involving pharmacological agents will also rely on these gene expression patterns. And in the long run, this approach will lead to, or at least that's what we hope for, will lead to so-called personalized medicine when, in ideal case scenario, you have every single patient analyzed for his or her expression pattern in response to a specific pharmacological agent. And then once you contract a condition that can be treated pharmacologically, you go to the doctor, they look at your gene expression profile, they look at what genes you have, how active they are in response to certain chemicals. And based on that, they come up with like ideal schedule for you, what kind of drugs you should be taking specifically because of your specific genotype and expression profile of that genotype, and which drugs you should stay away from. A lot of, again, we'll talk about this when we talk about the SNPs, a lot of side effects, a lot of people dying each year all over the world because of the drugs, because of unexpected reactions to drugs that they are prescribed. And that would be it for this lecture. It will be, like I promised, a pretty quick one. Start studying the last two slides here. These are just for your self-study. They are not really adding any new information on top of what we already discussed. To go over these online learning modules, they can help you visualize things that are happening in the lab, as well as this one here. I think it's a pretty good one. So start studying for the second midterm. I'll update that information. A lot of people asking. And start also studying the files that I posted for the final exam. Some short reads, some short videos on synthetic biology, on cloning and things like that. So all of that will go towards the final exam. It's reflected in the name of the folder. So that would be it, and I'll see you in the next video.